Hi, everyone. It's a great pleasure to be here. I recognize a lot of faces, obviously. I arrived at the University of Melbourne about 6.5 years ago now. And I think last time I presented at WEHI was about 6.5 years ago. So it's a great opportunity for me also to update you on some of the work that we've done with my team. I'm the director of Melbourne Integrative Genomics. We are just across the road. It's a cross-disciplinary initiative at the interface between biology and mathematics and statistics. So we have a lot of things in common, and that's why I know a lot of you here. We are hosted by the School of Math and Stats. We have about 45 members, and we're also part of 2 ARC Centers of Excellence. One is MAXIS, which is Modeling Biological Systems. It's led by Michael Stumpf. And one is in Quantum Biotechnology. I'm part of that initiative, and it's led by UQ. Feel free to have a look at our website because we run a lot of computer analytical data analysis workshops. We have a monthly seminar series, we have reading groups, and a lot of activities there. So my talk is going to be about integration and integration of bulk and single cell and multiomics. I don't think I need to talk about the fruit and the smoothie and the tart here. So I'm just going to jump directly into the what do we want to do and why is what is single cell annotation still a big problem, and what do people do in that field, and how we propose to address this issue. So we're mainly interested in annotation of single cells, and often people use a reference single cell RNA-seq data set, and then they're going to query their own data sets onto that reference in order to annotate their cells. So there's different types of techniques, from projection, prediction, data integration. What we need to do this task is a high quality reference data set, but also once you have this, you need also effective analytical methods. And so we've been working a lot with Christine Wells, trying to first establish what a reference data set may look like, especially if we start from bulk data. And of course, back then, that was 4 or 5 years ago, I was really questioning why we need those atlases from bulk. We had access to a large number of bulk data sets that are hosted in her database, Bioinformatics. And so we wanted to increase the sample size. Another motivation is also we wanted to include data from the community. So try to combat a bit this data obsolescence, especially as we were moving towards single cell. And from those atlases, if they're of high quality, the question was also, are we able to identify signal from noise? Are we able to benchmark studies against that reference atlas? And then finally, can we predict the phenotypes, whether of single cell or bulk samples, based on this atlas? And so you will see how once we establish some of those atlases, we then realized that we needed new projection methods, new prediction, data integration methods, as well as visualization. So visualization is actually quite key for this kind of work. So this is the outline of my talk. I'm first going to talk about some preliminary work that we've done. And then the last part of my talk is going to be about this new method that has been developed by my postdoc, Jadon, who's at the back there, and show you some case studies of that new work. So with Christine, we built different atlases, including this is a bulk blood atlas. Basically, it's a combination of 38 studies from 900 bulk samples coming from 14 RNA-seq platforms. In stem cells and in bulk, the phenotype is known. So we had this information. And then once we managed to collate this data, we can visualize it, for example, in a simple principal component analysis plot. So this is a 3D PCA, and each dot represents a bulk sample. And the color represents the non-cell type of those samples. So you can see this is a nice atlas. And once you've done that, then you can query any new study you have. So this is why I call it a query. This is a query bulk data set where basically you calculate the predicted component of those query samples based on the atlas you've learned before. And so, and then you can basically visualize, based on those predicted components of the query, you can then visualize them on the atlas. And so, for example, in our atlas, we knew that the first component was explaining the progenitor versus the mature cells on the first axis, and on the second component, it was more the sample source. And so we could see that the induced pluripotent stem cells that are in vitro progenitors were projected around here, near the progenitors and near the in vitro cells, whereas those ones that are dendritic cells were projected near the in vivo cells that are and mature cells. So this is just a validate. Basically, this is how you can benchmark your own data by saying, oh yeah, that's exactly what we were expecting on that atlas. Those of you who have tried to combine multiple data sets from different platforms know that it's actually not an easy task. We needed to do a lot of processing. So first we needed to select the data sets so that they're of high quality. Here, the different symbols represent different platforms, different RNA-seq platforms. And so once you try to combine them into a PCA plot, all you're going to see is often the platform effect, because it's much, much stronger than the biological effect. And so we did, one thing we did is we normalized further the data so that every study or every data set is comparable to each other. So the expression levels of a given gene is comparable between data sets. So we use a rank transform. And then in order to remove this very large platform variation, we did an extensive gene filtering, where basically using a variance partition method, we can estimate, what is the variation due to platform or to biology, and do we remove the genes that have a strong platform effect, so we only retain the one that have a biological effect. And this is how you can then obtain those atlases that have very little platform effect. So this is in collaboration with the team from Christine Wells. I'm just going to give you a bit more insight about this gene filtering step. On that plot, you have about 13,000 genes in rows. So each line represents one gene. And each line is colored according to how much residual variation they explain, how much platform variation they explain, or how much cell type variation they explain. And so we rank them according to cell type variation. And so you can see, on a collection of 13 or so thousand genes that were in common between all these data sets, a lot of them have a lot of platform variation. And so in the end, we had to keep only the first few 2,000 or 3,000 genes, because they're the ones who include the cell type variations, the biological variation of interest, and they don't have a lot of platform variation. So I'm showing you this, so you keep that in mind, because I go back to, do we actually need gene filtering when we build those references? And in the end, we, as I showed you before, we built those atlases. We created quite a few. And we then had this very nice visualization, in a PCA plot, where you could then project your query bulk data set on the atlases. And the next step was really to look into whether we could do the same now by projecting single cell data onto those bulk atlases and see whether we could, use that as an annotation tool. And of course, those of you who work in single cell, they know that, it's not just tweaking the method. You actually need to reinvent completely what existing methods, for example, to do this kind of work. And so this is the work we've done with Yidi, who's a PhD student at MIG, also supervised by Charny. We used the same pipeline as before. But for having a query that is a single cell, then we needed to, we proposed actually two approaches, which I'm going to briefly present. One is either when you start from single cell, you aggregate your single cell into pseudo bulk, or we impute the zeros. And then after that, we did pretty much the same thing as before. We project the query single cell onto the reference atlas, and we added a few more tools to refine a bit the identity prediction of those cells. So in Syncast, we have two options, as I said. The first is to do pseudobulk aggregation. It's quite common to do this nowadays, because we can then reuse the tools we have developed for bulk data, but using single cell. The idea is really when you aggregate your single cells into a pseudo bulk, you mimic the structure and the variation of bulk samples. We did that using Bootstrap. But of course, you can only aggregate the cells that are well clustered first. So that poses a problem when this doesn't happen. So the other option was to do this zero imputation. Basically, as I was saying, so either when your clusters are not well defined or there's a high variability, when the sparsity is high, so in single cell we have a lot of zeros, or, and that's quite important, when the identity of your cell is actually expected to be along a continuum. So this is not like it's not like, oh, you belong to this cluster or that cluster of cells. It's actually in between. And those of you who work in that area know that dropouts are a massive problem in single cell. And so one solution is to do zero imputation. And what we show in Syncast is that actually doing that avoids to do any batch correction or any library size normalization. So we extended a method that is graph-based, that is called Magic, and we extended that in Syncast. So let me show you an output of Syncast. This is a busy plot. The reference data set that we have is actually the blood atlas that we built before. And the reference is indicated in dots or in these big ellipses, and they're colored according to the known cell type of those cells. The query dataset is a single cell dataset of dendritic cells and monocytes, and each is represented with a cross. So each cross is a single cell. So you have basically, we have the atlas from Rajab, and then we overlay or we project the single cells at the cross. And I just wanted to show you that, for example, if we're interested in this dendritic cell called DC5, it was actually a new DC population. It's actually sitting between the plasmacytoid dendritic cells and the other DC cells. So it is actually along a continuum here. So this kind of plot already tells us a bit more about what is, what is the identity of those single cells. So this is very visual, and so you can have this intuition, but of course it's better also if you have some sort of a quantitative way of looking at those predictions. So we extended the KP-Barra method, which is a weighted restricted regression analysis. So here, each row here represents, each column, so each line, vertical line represents a cell. And each row would represent the known cell type from the reference. And so if we are interested in the DC5, they're in that red rectangle here. And you can see that they're mostly predicted as plasmacytoid cells, but also a little bit also with the conventional dendritic cells here. So this is a quantification way of looking at the annotation of cells. And so this is quite relevant to think about a continuous phenotype rather than this is a PDC or this is a conventional DC, to think more about continuum of those cells. So that leads to the next part of the talk, which is how we extended that concept of continuous phenotyping. So that was the first, I think, our first motivation. And the second motivation is, could we actually extend this kind of work, but looking also at different types of multi-omics in single cell? So not looking only at a single cell RNA-seq, but ATAC-seq and spatial transcriptomics and so on. Okay, and so this is where Jiadong proposed this new approach called Phi-space. And it starts very similar to any other tools that you may have in SORAT, for example, where you have a reference data set. So here we can do bulk. People don't do a lot of bulk here, but we use also bulk as a reference, or you can use single cell as your reference. What is different from other approaches is that we look at different types of phenotypes. We don't look only at the non-cell types. We may look at different sample sources or any information that we think is interesting that characterizes those experiments and those cells. So once you have chosen your reference, then you will query your data set. So for example, it can be a single cell data set. And so people do that by training on the reference and then predicting on the query. And we obtain the predicted phenotypes. Okay, so very standard, train, predict kind of framework, except that here we do not use a hard classification method. We use what we call a soft or a fuzzy classification method. So the phenotype is actually going to be a continuous value. And the next, so this is what I highlight in blue, the novelty on how we tackle this problem. So we have this continuous phenotypic characterization of the query samples. And also another thing that is quite interesting here is that we then look at what we call this phenotype space, and we visualize it and we do some downstream analysis directly on it. And I will show you that it has some benefits in doing that. Okay, so how do we do this training and prediction? We use a PLS method, so it stands for partial least square regression. I show you a little bit what it means, but basically PLS is used to integrate the reference data set and the phenotype, and we extract the regression coefficients. From the regression coefficients, we can then predict on the query. So the query data set is here, and we can then obtain the prediction, the phenotype, the predicted phenotype of this query data. We also use it for the reference. We recalculate the predicted reference. Not that you really need that, but we want to use it then to do further visualization. The other thing I forgot to say is that in the PLS, once we have those regression coefficients and all those outputs from the PLS, we can also identify which are the genes that contribute to the prediction of each phenotype. So just remember here, our phenotype can be a specific cell type or can be a specific sample source. So it's very easy for us then to go back to what are the top genes that actually contribute to that prediction. And then we do an extra dimension reduction visualization here using PCA. So basically, because your phenotypes, you can have many phenotypes. You can have 60 cell types as well as, I don't know, 3 or 4 different sample source. So that's quite big. So we use PCA to reduce that phenotype space. And then we project the phenotype of the predicted phenotype of the query onto that space. And then we can actually have, and then we can visualize all of this into a plot. So most methods will use something quite similar. So I'm thinking, for example, of SORAT, where they do a training on the reference. And then they will do the prediction here, but instead of looking at the continuous phenotype, because they also get it, they will use a hard classification method on that. So instead of saying, oh, you're in between plasmacytoid dendritic cell and a dendritic cell, they will say, well, actually, you're a conventional dendritic cell. And also, they usually visualize the data in the expression space. They don't actually look at this phenotype space. And then once they do this classification or prediction, then they will color the cells according to the result of the hard classification prediction. So that's why you get a UMAP with different colors of dots that represent the different predicted cell types. Those of you who know a bit of my work know that we work a lot with partially square regression because it's quite an attractive method for big data, and it allows us to integrate different types of data sets. So classically, in my lab, we've used PLS mostly to integrate different omics. So you would integrate gene expression data and, I don't know, proteomics or metabolomics data. And the way this method is working is that it actually decomposes these data matrices into a set of vectors. So you have the components and you have the loading vectors, so that the covariation or the correlation patterns between those two data sets is extracted. And then you have also other byproducts such as the regression coefficient matrix and so on. In our case here, we use the reference data set and we use the phenotype in order to understand and to model the relationship between the phenotype, the expression data set, and the phenotype. And so typically in PLS, that's what we do. We integrate, we extract the components, we visualize the components from either the X data set or the Y data set into a sample plot. We do a bit of variable selection, and we're pretty much done. Whereas I think what has changed here in this type of problem is that we're actually looking at prediction a lot. So not only we learn from the reference and the phenotype, but then we have this query that we need to predict. And then we add additional dimension reduction visualization in order to make sense of this prediction. So that's also what is a bit different from the work we've done before. Okay, so I'm going to move on to 3 different case studies just to show a bit of the breadth of application of Phi-space. So first, I'm going to show you how we can annotate single-cell RNA-seq from a bulk RNA-seq reference. Then I'm going to show you how we can do the same by annotating single-cell ATAC-seq from single-cell RNA-seq. And finally, I move on to spatial transcriptomics. Okay, so the first example is a single-cell RNA-seq data set from dendritic cells again. But it's a different query. And the reference we have is bulk RNA-seq. And so one thing that I want to show you here is first, what would happen if you look at the expression space? So just the RNA-seq data with a single cell bulk, and you just try to put them all together. So the bulk samples are the dots, as usual. And they're colored according to what we already know about those cells. So here they would be either DC1 cells or other type of cells. This is exactly the same plot on the right, but this time I show you the sample source. So whether it's in vivo or in vitro. The single cells are the crosses again, and you can see that there's nice clusters. So day 9 is the fibroblasts that have been cultured for 9 days. And then you have all different days. So the clusters of the single cell themselves look good. But when you try to integrate both type of information and you look at the expression space, you can see that there's still a massive, well, you can call it a platform effect. between the bulk data and the single cell data. It's very common. I mean, here we haven't even imputed the zeros or anything. So of course the single cell are going to be all in the middle, whereas the bulk data have much more variability. So if we then look at the phenotype space, which I've been talking a lot about, this is what you can see. Basically, the batch effect is removed between those two types of data. So we can start to see, for example, that the day 9 samples here, they are very similar to the DC1 bulk samples here. And then because we have integrated all different types of phenotypes, so cell type, but also sample source, then we can also see that the day 9 samples are also clustered towards the in vitro bulk samples. So this is how you can get a bit more information about the phenotype of those cells and how this batch effect has been removed. The other thing is you can extract, for example, just that plot here, just extracting the single cell, and we can then overlay the phenotypes on it on a biplot. And the phenotypes belong to the reference data, so the bulk data. And so this is exactly what I told you before, that the day-night samples are more in vitro-like, and so on. So you get more information about what is the link between those cells and their likely phenotype, as well as what is the correlation or the association between those phenotypes. For example, I can say that the DC1 from the reference are kind of close to the in vivo samples. So you get all that information, so both on the phenotypes as well as the cells. So I'm going to dig more into this thing, but I want to show you what's the benefit also of doing some variable selection here. So this is just looking at the reference, and the reference is the bulk data set. And this is what you would get if you do, for example, a PCA plot on all the genes. So at the top here, the colors represent the platform. In the middle, this is the cell type. In the middle of the cell type, and at the bottom, the sample source. So you can say, as I was saying before with the work with Christine, if you take all the genes, you're going to have a massive platform effect. This is what we did with Bioinformatics, where we filtered the genes so that we would remove the genes with the platform effect. So you can see now there's less of a platform effect. But as a result, for example, the sample source, is very mixed up here. And with Phi-space, we can also, focus only on the most important genes that explain, for example, both the cell type and the sample source. It does keep a bit of the platform effect, but the reason why it does this is because it is partly confounded with the sample source. And the sample source is still very important for us. So this is what is happening here. And so in the end, the take home is that we don't drastically filter the genes anymore. We keep the information about the genes that we think would tell us a lot about the sample source here or the cell type. And then you can dig more into what are the top genes that contribute to, for example, a specific sample source or cell type. So this is from the reference. We can identify this with the PLS. And these are the top 20 genes from the bulk reference. This is the expression levels of the top 20 here on the x-axis. And only for those who contribute to the in vitro phenotype. So this is on the left-hand side. And this is the same 20 genes, but this time I show you the expression levels in the in vivo bulk. So of course, in vivo, what is good is that in vivo, nothing much happens, which really confirms that those genes are characterizing the in vitro more than the in vivo genes. So this is in the bulk reference, and then you can have a look at the expression of those genes in the single cell query. It's not as neat because, the single cell data have a lot of zeros. So now the box plots look a bit weird. But basically, what we think, from what I showed you before, we think that the single cell at day 9 are very similar to the in vitro. And so you can see actually that the trends are quite similar to the bulk reference. So that really confirms that we picked up similar patterns between those two data sets. And that it's much more noisy or close to zero for a lot for the in vivo data. So that's it for the single-cell RNA-seq annotation based on using a reference bulk RNA-seq data and looking at the phenotype space. You can also do some gene ontology. For example, this is the gene ontology of the top 100 genes that contributed to in vitro. And so we actually saw that it, identifying these genes makes sense. So this is why, we've been arguing that, it's good to look at the phenotype space because we get more insights into the phenotype of the cells, but you can also identify, okay, what are the genes that are actually doing something here? I'm going to move to the second case study, which is called bridge integration. So this is when you have a reference omic data set, for example, single-cell RNA-seq, but then your query is a single-cell ATAC-seq, so it has nothing to do with single-cell RNA-seq, but you still want to annotate your cells from a single-cell ATAC-seq. And so we use the concept that is being proposed by the SORAT team in SORAT V5, of using a bridge. And the bridge is basically a multi-omics of single-cell RNA-seq and single-cell ATAC-seq that is measured on the same cells. So we use similar principles to them, but we use our tool with the PLS to do that. It's a bit long to do because what you do is you first train your reference single-cell RNA-seq with the phenotype. Then you query the single-cell RNA-seq onto that reference in order to get the predicted phenotype. And then you relearn or you retrain another classifier based on the single-cell ATAC-seq of the bridge with the predicted phenotype. And now you can bridge, and then you can query your single-cell ATAC-seq on the bridge in order to get your annotated single-cell ATAC-seq. So there's a bit of some steps to do here. Here we looked at a reference that is a bone marrow mononuclear cells in single-cell RNA-seq. So this is the first line here. The bridge is a single-cell RNA and ATAC-seq. There's actually 12 batches in those data. So we take only one batch as a bridge. And then we can look at the single-cell ATAC-seq as queries for the other 12 batches, only looking at the single-cell ATAC-seq. And we have access to the annotation of the single-cell ATAC-seq because we have the single-cell RNA-seq equivalent on those same cells. So we can have a look, just a simple UMAP plot of, what would happen if you take 3 batches of this single-cell ATAC-seq. And of course, you would see the colors here represent the different batches. And of course, you would see, some strong batch effect here. Whereas with Phi-space, if we, look at the predicted phenotype of the single-cell ATAC-seq, and we overlay them, we can see that there is no batch effect. So just to reiterate again that we are able to then remove this batch effect between the reference and the queries. We can have a look at how good is the method at clustering on the single-cell ATAC-seq. The first row here represents the clustering based on the major cell types. And the second row is the clustering based on the fine lab label. So you have 22. We use, as I said, one batch as a bimodal bridge. And then we query the single-cell ATAC-seq 12 times on that bridge in order to cluster the single-cell ATAC-seq. Once we get the embedding, which are the reduced dimension of the single-cell ataxic that have been predicted, then you can run, for example, a k-means. So we compare our approach, we compare Seurat bridge, we compare a simple PCA that is run either on the ataxic peaks that have been aggregated at the gene level or the peaks themselves. And just to show you that generally, both Phi-space and Seurat are pretty good for clustering the single-cell ataxic. For all these different types of measures. So you have the RAND index, the mutual information, and so on. The purity, I think, is where actually our methods is quite challenged. So this is for the clustering on single-cell ataxic. You can also have a look at the hard classification if you want, because this is what the other tools are doing. And so what we do is we extract the continuous predicted phenotype of Phi-space, but then we apply a hard classification on that. And even with that, we can, our classification error rate is actually quite good and quite competitive to Seurat. So this was just an example of showing you how we can use this bridge integration now to annotate other omics based on a reference that is different, And I'm going to move on now to the next case study, which is spatial transcriptomics. So I don't think I need to introduce too much also about spatial transcriptomics, but here we use a reference from Azimuth. It's a human lung atlas. And the query is a small area of lung cancer tissue. So this is how the cells, so each dot here represents a cell, because this is using nanostring cosmics. So each dot represents a cell. It's been annotated by the authors into a different spatial niche. If we use Phi-space, so we learn from the reference, and then we try to predict what is the likely identity of each of those cells. For example, for a very particular phenotype, here that would be AT1, which is alveolar type 1. And this is what you get. You get that map where basically each dot is colored according to this continuous phenotype. Just to put that into what would happen if you use a hard classification such as Seurat, you would get basically a few red dots where it says, yes, it's AT1. Actually, you don't see that many. I don't think you can see much here. So that just shows you the benefit of using this continuous prediction rather than this hard classification. So as I was saying, you can do that for every phenotype. So basically what we do is we look at the predicted phenotype of the query for each of those phenotype labels. And you can do that we have 61 cell types labels here. And then you can color all your cells according. So this is the B cells, which are the tumor cells. The CD4 T cells are here. So you can look at each of these cell type labels individually by looking basically at that. Or as I was saying before, you can combine or reduce the dimension of all these prediction in this step 3 here. And you would obtain this kind of map. Where basically you can have a better insight about all cell types combined. Do they reflect more the tumor side of those cells or the non-tumor? And the reason why we can do this is because we can look at the phenotype and how correlated they are or not from the reference. And so, for example, the cells here that are colored with in blue are actually the cells that have a high identity of those cell types. So you can start to disentangle a little bit and what's going on when you try to predict the phenotype of the cells. And that's much more insightful, we think, than looking at a hard classification. I think that's it. So we propose to use continuous phenotyping rather than based on soft classification. And so we use PLS as a soft classifier, but we can use any classifier you want. What we like is that we propose this new type of visualization of the phenotype rather than looking directly at the expression space. And that allows us to get more insights into the analysis, how similar the cell types are. But we can also have a look at, what are the key genes that contribute to each phenotype, and so on. By doing this kind of approach, we address the issue of platform variation without having to correct for batch effects, because we work on these projections. We work into another space, which is the phenotype space. It's much less noisy. And so it allows us to integrate bulk and single cell. It allows us to do mosaic bridge integration and so on. And so this work hasn't been published yet, but we're very happy to share it because I think it has a lot of potential. So if you're interested in, applying it or testing it, we're happy to work with you through that. Jiadong is going to focus a lot on the spatial transcriptomics. We are testing different types of platforms, but we also want to test it in cancer. And this is the team, but I really want to thank Jiadong for all the hard work he's been doing in the past 18 months, as well as the whole team, and also people in Mixomics who continue to help improving the tool. Thank you.
Thanks, Kim, for your wonderful talk. So questions?
Hi, thank you for the talk. I have just one question from the third case study on the spatial transcriptomics data. I'm just wondering, how was the spatial information incorporated into the query? Is it like used as part of the phenotype information?
No, okay. Currently it's not. I mean, I might be wrong, but currently we just use it as the, just as a visualization. But that's something we could, I don't know if that would make sense to actually use it as a phenotype at this stage. It depends on the resolution, I think,
Golden has a question online. So do Phi-space and the reference data set help with identifying novel cell types in single cell data? Can you see that a cluster in a new data set is novel?
I think visually you can have this intuition. I'm just trying to, so the reference will always be very well characterized because that's the aim of a reference. But you can start to have a visual intuition if you can start to see that there are cells that are not actually sitting on a particular non-cell type or another one. So that's the example that I showed here for the DC5. It's really in between, non-cell types. I think from there on, you need to do more downstream analysis here. Have a look at the expression of the top genes. For example, that are highly expressed in this group of cells or something like this, in order to kind of characterize a bit more, what might be this new type of cell. You will also probably use this kind of outputs like that, just the phenotypes, and see how it fits, with the different phenotypes. So, for example, here, we can say that, there might be some cells in the middle and they're actually very close to DC1, but they're a bit in vitro or they're a bit in vivo. So you can start to kind of characterize that. So it really depends also on how much phenotypes you want to include when you do the training. Yeah.
Oh, thanks. Ramil has another question. You have mentioned that there is confounding effects between batch and biology in the STEM formatic data. So how did you stratify genes based on platform effects and biological effects for gene filtering?
Yeah, so I think it's, so in STEM formatics, we said we don't want to have any platform effects. So we were pretty ruthless and just remove all of them. in the hope that, the one that would remain would have an interesting biological variation. Whereas I think in our case, we say, well, actually, there is this confounding that we cannot remove. We're still interested in the sample source anyway, so we have to kind of balance it. But what I've shown after that is that actually the method is pretty good, is pretty robust against the platform effect anyway. So it will still focus, I think, on the gene that has the most biological variation of interest.
Great talk. For the analysis of the single cell ataxic data, how does that compare with using, I don't know if that's possible to use ataxic bulk as a reference for the single cell attack?
Yeah.
And my second question is it possible to quantify how much information is coming from single cell RNA-seq and how much is coming from the single cell ataxic of the bridge data set?
Okay. First question, yes, I think you can. We haven't tried. I don't know if there are any good reference bulk ataxic that we could use. So it's more, the work on, what is the best reference we can use. For the second question, I think we have, okay, so here for the bridge, we have the annotation of those cells based on the single cell RNA-seq already. So you will be able to compare, is this prediction similar to what we expect, I think. So you can say, oh yeah, it's very close to RNA-seq, to what we knew from the RNA-seq side. I think for the ataxic, it will be, you can do the same as well, because you also have basically you have the annotation of all that bridge. And so you can start to, I think, have an intuition about, whether it's you in the right direction or not. And in this case study, we have, they are actually bimodal. So all the ataxic are actually bimodal. We're just making it a bit fake here. So we do have the annotation based on the single cell RNA-seq. So there might be a way, we haven't really thought about this on how we can quantify it and say, oh, actually, it's better to use single cell RNA-seq than ataxic for the annotation.
I have a question. Thanks for the lovely talk, Kim-Ann. I was just wondering, with the last case study with the spatial data, the sort of strength of the Phi-space thing. So I guess it ranges from 0 to 1, or
it can be negative too.
It can be negative as well. So does that correspond to like how confident you are on that prediction? So like a darker red, you'd feel more confident, or is it sort of relative? So I guess here it doesn't ever really get to red, but it's sort of 0.5.
Yeah.
But you would still say those 0.5 colored blobs are
so
more
it's,
likely 81.
Yeah, it's similar to some sort of it's a score between 0 and 1. So 1 is like, yeah, you bang on 81, and 0 is like, no, actually no. So in between. So I think it is similar to some sort of confidence or likelihood of what the cell is likely to be. And so if you have here, basically it says a lot of no, and quite a bit of maybe yes. And so what is interesting is you could say, well, it is actually a mixture of 81 and other things.
So I think what's tricky as well is that, like, this imaging-based spatial transcriptomics, the data generation mechanisms are really different to like a single cell RNA-seq based reference. And I noticed with all of the cell types that we never really have a lot of very confident continuous classifications. And I'm sort of wondering if even using a single cell as a reference, is it the best way to go? And is that sort of highlighted here that we're not really getting like super confident, but we're getting a sort of good idea maybe of what
I think we
are
getting close. We might be able to have reference spatial data at some stage. And maybe we can do that as a reference rather than, yeah.
And I'm just, sorry, I don't know how to interpret the right-hand plot. Sorry, I know I'm asking lots of questions.
So
but
the right-hand plot is actually that part here. So basically you have a prediction for each cell type. So we're sitting here. So each column represents for each cell, how likely it is to be a T1 or B cell or blah, blah. And then you can then combine all these predicted phenotypes into one single score. And so that score is going to be weighted according to, is a B cell very similar to a T cell and all these things. So this is what I was showing you here, is that actually the cells that have a strong negative score at the end will be a combination of those different cell types. And so then you need to have a look at, are they, tumor cells, or are they, similar to tumor cells or originating from tumor cells?
Thank you.
Hi, cool talk. Could you just go back to the previous slide, John? It's really interesting because it seems like you can be very flexible with like what you choose your phenotypes to be as an input. If you like dropped out one of those phenotypes from the reference, how much does it change? Or do you think would it change much what happens in the output?
Not drastically, because I think a lot of those phenotypes are actually correlated. So it may lean more towards, say, B cell rather than CD4 T cell, right? If you're removing, yeah. So I think as long as you have enough information there, that's fine. But I think that relates a bit to the question from Gordon, that, can you detect some continuous thing? In the case of spatial, we might be able to only infer what we've been missing, but of course it will be, a bit hard. Yeah.
Thank you.
So sorry, we are running out of time. So please join me to thank Kim for the wonderful talk.